Okay, could you please do me a favor and turn the lights on back there? I had a question. What's your question?
Because it represents in different directions.
Yeah, 1 second, I'll be right back. Zach, just turn the light on. Okay. This is going back a slide to review a little bit. Watson and Crick. Which, remember, who are Watson and Crick?
They invented
They didn't necessarily steal, they just didn't give her credit.
No,
Okay? But they did use her work, they did contribute, because they did use her work to come up with the structure of DNA, but it was not them that actually took the images, the X-ray diffraction photos of the DNA. Yeah.
So can I use your card? I've never had this right now.
You should have it written down already.
Alright, just wondering.
And their model served to tell us that DNA controlled cell function by serving as a template for protein structure. So that's what DNA does. It controls what proteins we have in our body. We discussed that. And the way it does that is by sections of 3 nucleotides. The 3 nucleotides are those complementary bases. But the base is only 1/3 of the nucleotide. The nucleotide has what 3 things to it?
Sugar, nitrogen, base, and
Phosphate group. And the phosphate group. Those 3 things, yes. And so, but since everything else is the same, what we're really concerned about with our nucleotides is what type of nitrogenous base they have. And when we have 3 of these together, that makes a triplet, or what we call a codon. Have you heard the word codon before in science?
No.
Okay. Well, then that'll be something new. We're going to learn about codons. Each codon, or each set of 3 nucleotides, is the instructions for making 1 amino acid. So do not be confused the difference between nucleic acids and amino acids. Nucleic acids are the building blocks of DNA, deoxyribonucleic acid. Amino acids are what the DNA is coding for, those triplets, those codons. Each one, there are 20 different amino acids that are possible, and when we make amino acids, they come together to make proteins. So the building blocks of DNA are what? The building blocks of DNA are nucleic acids, deoxyribonucleic acids. So the building blocks of DNA are nucleic acids. The building blocks of amino acids are what makes up amino acids? Nucleotides, 3 of them. And then what makes up proteins? Dion? What makes up proteins? Amino acids. Thank you. So that's how they all come together. Nucleic acid, DNA. Okay.
Did we already write this?
I believe so.
Oh.
Nucleotides go to amino acids, and amino acids build protein, and protein builds other structures in your body. Okay. This is the process of DNA transcription, what you should have read about, what we just took a quiz over.
Yep.
Your assignment was reading about DNA transcription. There's 2 parts to replicating DNA. The first part is transcription, and the second part is translation. 2 parts. Can you guys see okay, or do I need to move out of the way? See? Okay. DNA can unzip itself. What does that mean when I say unzip? Take apart what? The 2 sides of the double helix, which we call, from your quiz, complementary strands. So unzipping means separating the two complementary strands of the DNA double helix. So that's unzipping itself. And then RNA nucleotides match up to the DNA strand. Notice, this is important, and this is new, so you can start writing this down. It is not DNA nucleotides that originally match up. It is an RNA. Where does RNA come from? Well, in a roundabout way. Where does, those of you that study the organelles, where do we get RNA? What'd you say? Ribosomes, right? There's three types. There's rRNA, mRNA, and tRNA. Right now we're talking about rRNA. Okay? And it's going to match up with a nucleotide, and by that we mean that that's where you guys did, if I said that I unzipped the double helix, and I said that I had one nucleotide that was had an A base. What RNA nucleotide are you going to pair with that? T. If I had the base on my nucleotide was C, what RNA nucleotide are you going to pair with that? There you go. So you get the idea. But it's not officially matched. It's not officially a matching set yet because we're using RNA. We're using an RNA template. We're not using DNA. Both DNA and RNA are formed from nucleotides, and they're both called nucleic acids because it's deoxyribonucleic acid, deoxyribonucleic acid, and ribonucleic acid. So they both are types of nucleic acids. Oh, sorry, I misspoke. It's mRNA, not rRNA. mRNA. Messenger RNA is what that stands for. We'll have notes of that later, but.
We should have started next
For your birthday? Yeah.
Today's Sean's birthday.
We should have.
Happy birthday!
Oh, I'll sing like Sean next Friday.
Tell him.
Happy birthday to you. Happy birthday to you. Happy birthday, dear Sean. Happy birthday to you. Okay, this gives you a description of what's taking place. Okay? The reasoning, this is important. The reason that we are using mRNA to duplicate our DNA at this point in time, instead of just using DNA, why do we use that excess step? Is because we have to, this is taking place in the nucleus, but eventually this has to go outside the nucleus. And the DNA has to stay in the nucleus, so we need something that's capable of moving outside the nucleus. So that's why we are going to do it this way. Okay, yeah. DNA translation, the second set, the cell uses information from messenger RNA, mRNA, to produce proteins. Yes, there's just a picture.
What?
What up?
I was like talking to you. I was like, she thought I said stop.
Here is a good description or a good image that shows this. I'm going to explain this image, but we will have separate notes over the specifics of DNA translation. But here is your RNA strand, your complementary RNA strand that we copied from the DNA that was unzipped by
Helicase.
Helicase. Helicase unzipped it. What's the enzyme that added these RNA complementary parts?
Right.
What?
DNA polymerase.
DNA polymerase is the one that added the RNA part. Once it's transferred out, it's going to find a ribosome. Ribosomes are where we're going to put together or build our proteins. In order to build a protein, what things are we using to put together? What kind?
Nucleic, ribonucleic acid. Amino acid.
Amino acids. Amino acids are what we're going to put together to make a protein. So that means we need to figure out what amino acids we need to use to build this protein. We need to follow our instruction manual. Okay? Our instruction manual has sets of threes. We call these sets of threes what? Triplets or codons. Okay. This strand is going to find a ribosome, or rather a ribosome is going to find this strand outside of the nucleus. And the ribosome, to the ribosome comes these little things right here. If you had to pick a letter, what does this kind of look like to you? T. So it is the tRNA, or transfer RNA. The tRNA is going to come to the ribosome. We call the ribosome the site for protein production. That means it's just a location. It's not doing any of the work. It's the location that it takes place. So it's sort of holding the RNA in place so all of the parts can come together at a location. The tRNA, notice what is connected to the tRNA on this side? Nucleotides that are going to match up. So each tRNA has a set of 3 nucleotides that are going to match up with our RNA strand. So it's matching them in sets of 3. What's on the other end of this tRNA is a circle. This circle is representing a type of amino acid. So every amino acid matches a specific codon. So when this tRNA comes to match up with this codon on the mRNA, then it's going to build the protein by a set of amino acids. If that is still unclear to you, that's perfectly fine because we're going to keep going over. I'll have notes over this, but I'm just trying to explain this particular slide. And some of you might catch on a little quicker than others, but this tRNA has a set that matches this up so that we get the right amino acid for whatever our codon is specifying. So that's how our instructions are working. Our instructions are going to match up with a particular amino acid. And then eventually these amino acids will get put together and they'll make one globular protein. These amino acids, it's overly simplified. These amino acids are shown as little circles. Amino acids are bigger than nucleotides. Okay, why is transcription necessary?
Because
It has to be right.
Yes?
So you form a new form of, or new strand of DNA.
Okay, but specifically, we have transcription and translation. Why do we need transcription? Why can't we just copy the DNA right there? Why do we have to first match it with mRNA?
Because we need a way to match it up on the other strand. So we need that.
And what did you say?
Need to match that with DNA strands.
Okay, Riley? Why? What's one reason that we need mRNA? Why can't we just match the DNA with the DNA strand right away?
There's probably a reason for it. I'm not sure.
well, there is a reason for it.
Okay. We'll see what your answer is. What is? Describe what is transcription. We just did.
We just did.
What's transcription?
DNA transcription.
Morgan, what do you have down for transcription?
Um,